The Homeobox sequence, which was shown by dark blue box, this sequence code for a specific sequence of 160 of 60 amino acid. As I have already told you that this Homeobox sequence is 180 base pairs. So this 180 base pair corresponds to 60 amino acid. That is an amino acid or a protein that has got 60 amino acid. So we can say that the Homeobox sequence code for a specific sequence of 60 amino acid within the synthesized protein, and this 60 amino acid is called the homeodomain. As you can see with the dark green single line, it represents the Homeobox gene. A small square, it represents the Homeobox, and the Homeobox gene has synthesized, you can see the protein, which is somewhat curved, zigzag in appearance, and within this gene there is a 180 base pair coded gene, which is 60 amino acid, shown by 3 cylinders. So these 3 cylinders, they are called the homeodomain. The homeodomain sequence, as you can see in this picture, the homeodomain sequence or protein is shown by number 1, 2, 3. So the homeodomain sequence fold into specific shape consisting of 3 alpha helix. So these 3 cylinders, what their conformation is? Their conformation is in form of 3 alpha helix. So this specific region is called the homeodomain. It is a protein of 60 amino acid. Here the structure or the conformation the protein has acquired is called, in the homeodomain or for the homeodomain, is called helix turn helix. So there is a helix represented by 2, then there is a turn, and there is again a helix represented by alpha helix 3. So this conformation of the Homeobox protein having specific conserved sequence called the homeodomain, and this homeodomain has the conformation of helix turn helix, where the helix number 2 is there, it turns and joins with helix number 3, and in between them there is a short loop of amino acid. So with this particular structure, now this protein binds at specific region in the DNA. The helix turn helix shape allow the protein to bind to DNA and regulate the transcription of nearby genes. So it is by these helix turn helix conformation of the protein that the homeobox protein binds with certain region in the DNA, and they help in upregulation or downregulation, or we can say they act as enhancers or repressor in transcription for other genes. So we can say that these are the protein that contains the homeodomain are the transcription factor. The protein synthesized by the Homeobox gene contain homeodomain, and therefore they act or behave as the transcription factor, helping the enhancement of DNA, that is activation of certain region in DNA or repressing certain region in DNA. So these sequences are conserved, and because of them the similarity in the organization of animals are seen. So Hox gene are homeobox gene, and this Hox gene is found only in animal. Hox genes are responsible for the structural organization of the animal. The Hox gene sends information in form of proteins so that the ball of cells or the sphere of cell developing embryo, it forms the head region, thoracic region, abdominal region, then forms the segments, then forms the appendages. So these Hox genes are responsible for giving instruction for the organization of the body, structural organization of the body. So what are Hox gene? We can say that Hox genes are the subset of homeobox gene. There are homeotic genes which uh, drive the structural, or we can say that which uh, give instructions for the formation of various structure of an animal. Then the homeobox genes are the subset of homeotic genes, and Hox genes are the subset of homeobox gene. Hox genes are involved in the correct positioning of the body part in the organism, like where the head region will form, where the legs will be formed, where the abdomen will be formed. The Hox genes are found in all bilateral animal. Bilateral animal means all those animals which can be divided into two equal half when they are cut from the anterior to posterior end. That is when they are cut vertically, and we get right half and the left half, or right side and left side. So those animals are called the bilateral animals, and this Hox gene which instruct for the formation of structural organization in animals. So Hox genes are found in all the bilateral animal, and it suggests that Hox gene has existed in the common ancestor of all the bilaterians. Several Hox genes are found next to each other on a chromosomes, and they are arranged in clusters. As you can see that in a DNA, several Hox genes are present, and these Hox genes, they give instructions for the structural formation in animals. In some animal lineage, including vertebrates, the Hox genes have been duplicated, and which has resulted in the multiple Hox cluster. As you can see that these are the genes, three chromosomes are represented which are laid down horizontally, like one red line present on the one chromosome, the second red line present on second chromosome, then there are orange line present on both first, second and third chromosome, the red line present on first, second and fourth chromosome. So there are four chromosomes represented here, and on these four chromosomes the Hox genes are present. So this is the one chromosome shown in this picture. Similarly, the other chromosomes are also shown here. 4 chromosomes are shown here, on which specific sequences called the Hox genes are present, and these Hox genes have been duplicated several times. You can see on first chromosome, red Hox gene has not duplicated, but the orange has duplicated 3 times. The yellow one has not duplicated, then the green one has duplicated 2 times, the blue one has duplicated 4 times. Not only the genes are duplicated, but in different animals these could be deleted also. So the gap that is seen are those places from where the Hox gene has been deleted in organism. So here in this screen, 4 chromosomes are shown, on which linearly the Hox genes are arranged. When a Hox gene is mutated, body part end up developing in wrong place on wrong body, and these are called the homeotic mutation. As you can see in this example, in place of antenna, because of mutation in the antennal gene, antennal Hox gene, the leg has developed in place of antenna. Again, here the developmental pattern is shown, where the embryonic development is represented by this diagram, and different colors exhibited on this shows the different Hox genes which get uh, activated, and they form further formation of legs and detail in the embryo. The Hox gene are expressed in early embryonic development along with anterior posterior axis of the organism. So you can see that it is the chromosome is shown where Hox genes are present, and these Hox genes are responsible for specification in the growing embryo. The order of gene on the chromosome matches with the expression pattern along the embryo. As you can see, the first number of Hox gene is present in the first segment of the embryo. Then the other two are present also in the anterior region. Then the two Hox gene present in the middle region. Then again 3 Hox gene present in the posterior region. So the order of the gene on the chromosome is exact match to the expression pattern along the embryo, and it shows that the Hox genes have the spatial linearity. The spatial linearity is there. Along with it, the expression of gene also occur in temporal order, time-related order, starting with the expression of anterior Hox gene. So first gene which is present first will form the anterior region of the developing embryo, thus showing that not only the Hox genes are present collinearly on a chromosome, but they are expressed in temporal, that is time related order also. We know that there are the structural similarity. All the organisms, they have anterior region, posterior region, right region and left side. So the similarity is there because of the presence of the homeotic genes, or we can say for structural specification where segments will be formed and in which segment which appendage will be formed. So this type of instruction is given by the Hox gene. And what are Hox gene? Hox gene are the subset of the Homeobox gene. But similarity is there, but with the similarity, lot of differences is also seen in these animals, and why this difference is there we will discuss later. When each Hox gene is expressed, it encodes a specific Hox protein which act as a transcription factor. The transcription factor binds at a specific region of DNA and switches on specific set of genes in each segment. As you can see in this diagram, the Hox gene has uh, transcribed or translated the Hox protein, and this Hox protein has a specific 60 amino acid region which is called the homeodomain. This homeodomain has a conformation of helix turn helix uh, binding site, and with this conformation it binds at a specific region on certain uh, DNA region. This will enhance or switch on certain genes. Hence, it is said that the uh, product of Hox genes are the transcription factors, and this transcription factors bind at specific region on the DNA, or we can say on specific region of on a gene on each in each segment, and thus it causes the expression of certain genes. The transcription factor binds to specific regions of DNA and switches on specific set of gene in each segment. These activated genes help to promote the correct development of each body segment by regulating mitosis, apoptosis, and cell differentiation. So, to sum up, what we have uh, learned from the lecture, we have studied that Hox genes are the member of homeotic transcription family of genes that play a role in controlling the body plan along the craniocaudal axis or the anterior posterior axis, and they also help to specify the segment identity of tissue within the embryo. Drosophila, like all other insect, has 8 Hox genes. When we were discussing about the axis specification, I had told you that in Drosophila there are the maternal genes, then there are segment genes, and then there are homeotic genes. So these homeotic genes are the Hox genes, and in Drosophila and other insect there are 8 Hox genes. If you remember, I had told you that the homeotic gene has got two complexes, and these two complexes they have cluster of genes. The first complex was called the uh, bithoracic complex, and the other complex was called the antennapedia complex. Antennapedia complex has got 5 genes, 5 cluster of genes. If I name them, they are antennapedia genes, labial genes, proboscipedia, deformed genes, sex combed reduced, and bithoracic complex have the ultrabithorax gene and abdominal gene, abdominal A gene and abdominal B gene. So bithoracic complex has got 3 genes: ultrabithorax genes, abdominal A gene, abdominal B gene. That is, they are the 3 genes. And the antennapedia complex has got 5 genes called the labial, proboscipedia, deformed, sex chrome reduced, and antennapedia. So the 5 genes together, there are 8 Hox genes in Drosophila, which helps in the formation of segments as well as in the formation of the appendages. As you can see in this diagram, the Hox gene present on chromosome 3 in Drosophila, and they are arranged in linear order. First is the labial, then the proboscipedia, then the deformed, then the sex comb reduced, then antennapedia, and then there are the ultrabithorax, the abdominal A and abdominal B. All these Hox genes, they are shown in specific color so that we can know that where these genes in the Drosophila has been expressed.